next speaker. The next speaker is Marina Puchinskaya, and she's coming from the Minsk City Clinical Oncology Dispensary from Belarus, and I would give the word to you, please.
Good evening, dear ladies and gentlemen. Do you hear me well? Okay. Dear ladies and gentlemen, my name is Marina Puchinskaya. I am a clinician oncologist and also a pathologist scientist from Minsk City Clinical Oncology Dispensary, Minsk, Belarus, and it is my great pleasure to be here, and it is my great honor to speak at this conference. And in my talk, I would like to discuss cancer stem cell markers in prostate cancer. So, as we've already heard several of times during this day, sorry, I have no conflicts of interest. So, cancer stem cells are important for tumor development, progression, and treatment resistance. By now, the golden standard of the definition of CSC is functional assessment of two main functions: the ability to self-renew and the ability to differentiate into various cell populations of the tumor. And along with that, it is assumed that cancer stem cells are relatively rare and represent only a minor subpopulation of tumor cells. These facts inherently arise a question of how can we define cancer stem cells in tumor tissues or tumor cultures or anything like that. And so how can we distinguish them from any other tumor cells that we can see in our samples? So, and here we come to cancer stem cell markers and their importance. Cancer stem cell markers are molecules whose expression may be used to define CSCs in tumor populations, in patient samples, or in cell cultures. The number of CSC markers is very high and it's even growing. By now, there are more than 20 different molecules that have been used as CSC markers. You can see here only a short list of them. And in my talk, I will mostly focus on CD44 and CD133 as most widely used, and as my personal study is mostly devoted to those molecules. So, CD44, its synonym is PGP1, Li24, HCAM, leukocyte hormone receptor, H1, Hermes, and everything like that. Rather well-known molecule that was discovered very long ago, but for many years it was considered to be only an adhesion molecule, and its gene was mapped at 11p13. It contains 20 exons. It is a well-known cellular receptor of hyaluronan, collagen, laminin, fibronectin, osteopontin, and also some glycosaminoglycans. So it is mostly functions as a cell adhesion molecule interaction of cell with intercellular matrix. However, its functions also include gene transcription, paracrine signaling, contact inhibition, cytoskeleton remodeling, cell adhesion. This gene participates in inflammation, leukocyte homing, to lymphocytes differentiation. It is a fusogenic factor. It participates in cell migration and tumor metastasis and intercellular matrix modifications. More interestingly, it also participates in a number of signaling pathways, including oncogenic ones like EGFR, HER2, MET6, TGF-beta-R1 and TGF-beta-R2, VEGF receptor, Roger phases, RAS-MAP, PI3K at signaling pathways, and several others. It has also been well known that this molecule is highly expressed in different tissues, including in normal ones. And so it raised the question of how cancer stem cells that are seen to be rather rare in tissues may benefit from this highly expressed marker. The possible answer is the existence of CD44 isoforms. So in these pictures, you can see, first of all, how various signaling is connected to CD44 and its various isoforms, standard and virus, and also how those various isoforms are generated through alternative splicing, and also some post-translational modifications. Mostly studied is the so-called CD44 V6 that is thought to be highly tumorigenic and that is thought to exert most of the protumorigenic functions and pro-CSC functions of CD44. Another molecule, CD133, is much less studied. It was first described by Yin et al. In 1997 on hematopoietic stem cells. It is also known as AC133 and prominin-1. It belongs to a superfamily of prominins, including also prominin-2 and prominin-3. The gene was mapped at 4p15.3. The protein is 120 kilodaltons, and it is 5' transmembrane protein. The functions of this protein are less well understood, much less well understood than for CD44. Sorry. It is known that it interacts with cholesterol, and it is associated with membrane protrusions and some areas of membranes. But how it influences CD133 functions, and especially how it is connected with CC features, is not well understood. So, staining in tissues is variable, and it was described to be expressed on cellular membrane, especially luminal in lumina of different glands and also in cytoplasm. And it differs in different tumors. It is also thought that different patterns of expression may interchange in tumor progression, and that cytoplasmic expression has a worse prognosis than any else. And the numbers of CD133 positive cells are usually rather low, but they're highly variable. For example, its presence was described in from 1% to 62% of tumors, and in colorectal cancer, for example, it has been described to be present in from less than 1% of tumor cells to as high as 82% of tumor cells in a sample. Again, CD133 has multiple isoforms, mostly attributable to the presence of 5 different promoters that are active in different tissues and that generate protein isoforms of various lengths and various functions. It should be noted, it's an important notion, that in most cases, available antibodies recognize the AC133-1 epitope, the most well studied. However, AC133 epitope expression is not the same as CD133 expression, and if using antibodies to some different epitopes, its expression may be very different. So when we are talking about CD133, we should bear in mind that it is mostly, we are talking about AC133 epitope, but not the protein itself. Again, as each of the marker gives us the ability to enrich of the population of cells having cancer stem cell phenotype and cancer stem cell features. So it was a good idea of trying to combine those markers. As, for example, we have one marker defining one subpopulation and another marker defining another population. So maybe the combination of those markers will give us cells with better CHC properties than others. So most widely used combinations in the literature by now in various tumors are CD44, CD1 homocysteine, as we do, and CD44 high, CD24 low. It's a well-known combination for breast cancer, but also used in some other tumors. In literature, up to 4 different markers were used. For example, Lin minus, lineage minus, SK1 positive, CD48 high, TROP2 high in mice prostate. And it is also important to bear in mind that in human and in prostate, CC markers, even in the same organ, may vary. For example, in prostate gland, those phenotype was described in mice, but not in human. So, as CD44 and CD133 are important markers of cancer stem cells and are thought to be expressed on cells with CSC phenotype, and as cancer stem cells are very attractive target for treatment, it was a good idea to use CD44 and CD133 as treatment targets. By now, the results are mostly preclinical, and none of these strategies has entered wide clinical practice. But however, it was shown, for example, that targeting CD44 enhanced anti-tumor immune responses and cellular uptake of liposomal doxorubicin, a widely used chemotherapeutic agent. Against the anti-CD44 antibodies inhibited mTOR pathways, and so inhibited growth and proliferation of tumor cells. By now, various technological strategies are used to target CD44, like peptides, aptamers, celibinin, decoy receptors, and everything like that. Again, regarding CD133, as long ago as in 2008, the first works appeared on anti-CD133 antibody drug conjugate. Interestingly, with monomethyl auristatin F, why I focus on that, because this molecule, for example, monomethyl auristatin is widely used now, for example, in rituximab vedotin, a well-known and highly widely used drug that is already in clinical practice. So it's a very promising strategy. And those antibody drug conjugates effectively inhibited growth of hepatocellular carcinoma cell line in mice. However, this strategy again hasn't entered clinical practice yet, though again other strategies are used now. Anti-CD133 demonized targeted toxins, nanoparticles, bispecific T lymphocyte enhancer substances, bispecific NK enhancer substances, trispecific NK enhancer substances, aptamers, and anything like that. So I would like to refer to you to all those literature data that are really very intriguing and that show us that basic science and CSC theory may really change the future of clinical practice in some years. So when talking about CSCs in prostate cancer, the history of them began in 2001 in York, in Britain. So prostate CSCs are British in nature. In 2001, the laboratory of Norman Maitland first identified normal prostate stem cells using alpha-2 beta-1 integrin expression. In 2004, the same laboratory showed that CD133 is a prostate cancer stem cell marker, and in 2005, again, the same laboratory of Norman Maitland and Annie Collins described that the combination of CSC markers was used to describe prostate CSCs, CD44 positive, alpha-2 beta-1 integrin high, and CD133 positive. That combination is now considered like kind of classical, but usually integrin is not used nowadays, and in most recent works, CD44 positive, CD133 positive cells are considered CSC in prostate. When talking about prostate as a well-known example of hormonal-dependent organ, and prostate cancer is widely known to be hormonal-dependent, and hormonal treatment is widely used for prostate cancer treatment. Again, when talking about any biological process in prostate, it also raises a question of how those process is connected with hormones. So when talking about prostate cancer stem cells, they are androgen receptor negative. That was known for quite a long time, and androgen receptor negativity of normal prostate and prostate cancer stem cells was thought to be a mechanism of prostate regrowth in rodents after the well-known classical experiments of castration and reinduction of androgens. However, in experiments, it was shown that androgen receptor positive CD44 negative non-cancer stem cells can give rise to androgen receptor negative CD44 positive cancer stem cells. So that assumed to be a possible mechanism of escape from androgen dependence. Again, it should be mentioned that by now it is not well known what is the source of cancer stem cells, whether it is normal stem cells that are androgen receptor negative by definition, or the source of cancer stem cells are differentiated tumor cells that can then de-differentiate and change their transcription of various molecules. So this is a very, very interesting idea of how castration resistance can develop in prostate cancer. Another important issue is that androgen is not the only hormonal regulator of prostate cancer progression. Estrogen receptors are also considered very important, especially in the last decade, and it was very interesting that estrogen receptors were found to be expressed in cancer stem cells, CD44 positive, CD133 positive. So it again is assumed by Hu et al. In the rather old work of 2012 to be a possible mechanism of high prostate cancer risk in cases of neonatal estrogenization. So it is a well-known fact that neonatal exposure to estrogens may give rise to prostate cancer risk in adulthood, and possibly estrogen action on stem cells that are estrogen receptor positive may be an underlying mechanism of such condition. So in our own research, we use mostly tissues of prostate cancer from patients, so that is human tissues from real patients obtained during radical prostatectomy specimens. FFPE samples are stained using immunohistochemistry and double immunofluorescence. And we are trying to assess both staining patterns of those markers and their relation to some other characteristics of tumors, including clinical ones and including staining of androgen and estrogen receptors. The work is ongoing now, so by now I can tell you about our results of staining of CD44 and CD133. So here you see that on immunohistochemical staining in normal, non-cancerous prostate glands, CD44 is expressed mostly on basal lateral membranes. It is normal staining. It is well known that adhesion molecules can be stained like that. In prostate cancer, however, the staining is mostly lost. But in some senile cells, it can, however, be seen in this one, in these areas. So the expression of adhesion molecule is lost. It's logical. But again, if it is still present, it may be a hallmark of cancer stem cells. However, it can also be noted that in some cases, the expression is really high, even in cancer. And so we are using antibody to standard isoform. And again, it raises the question that possibly CD44V6 or any other isoforms may be responsible for this staining pattern. These are the first results of double immunofluorescence staining. So again, you see that CD44 is stained in normal cells, lost in cancerous cells, and CD133 is not present, at least in those samples that we have studied before. Again, CD133 staining, it was studied in endometrial cancer as a staining control and in prostate cancer. Here I used the figures of endometrial cancer as more like characteristics. So again, here we see those characteristic pattern of luminal membrane staining, and here see that luminal membranes are stained. Various intensity of staining can be observed, but however, this pattern of staining is thought to be rather characteristic of CD133, and it is also seen not in all the glands. And it is also very similar to that seen in other cancers. For example, one of the works also showed the same staining pattern in cholangiocarcinoma and in pancreatic ductal adenocarcinoma. So we think that maybe cells staining for both CD44 and CD133, they may be very, very rare in our practice, but they may represent those CSC cells that really matter in cancer progression. I should also mention some controversies of CSC markers that are known from literature and that can be understood by now. First of all, we should also be aware that CSC marker expression is not completely equal to CSC phenotype. By now, the golden standard again is functional characterization. And in many experiments, it has been shown that even when we enrich for example, CD44 positive, CD133 positive cells, even subpopulations that are negative may give rise to tumor. Maybe in more time, maybe in some other conditions, but still negative populations can also have some CSC properties. So we should also bear in mind that those CSC markers are very useful. They are not equal to CSC phenotype and characteristics. Again, abundance of markers. We know more than 2 dozens of various markers, and it also raises the question of whether cells expressing various markers have the same properties or they are heterogeneous. And maybe cells expressing different CSC markers may have different other properties, and maybe those cells that are identified by various markers may be of different value for tumor progression and of different stage at CSC hierarchy. Another important question is model system biases. It was shown, for example, it's well known that when we are using human tissues and human cell cultures, usually in experiments, they are transplanted into non-STID mice, and CSC properties are studied in those experiments. But when syngeneic mice tumors were used for such experiments, it was shown that even cells not expressing CCC markers can still give rise to tumors. So it arose a very, very important and unfortunately still unresolved question of maybe CCC markers do not choose for CSC cells themselves, but only for cells that are more able to grow in another immunocompromised host and in another animal. I think it's a very interesting question. And regarding prostate cancer, works on Trump mice prostate cancer model is very interesting, but it is a very important issue that we should always bear in mind. And again, we should also remember that CSC is not a cell subtype, it's not a like cell subpopulation, but it is rather a condition, a complex of some features and functional properties that may be changed during cancer differentiation, cancer dedifferentiation, cancer progression, and everything like that. So we should also think of it as, for example, regarding EMT, we should think of it as a kind of maybe even transient state, but not some well, characteristics that is inherently used in these cells. And so with all of that, I would like to rephrase a bit an important and well-known phrase by George Orwell that all cancer cells are malignant, but some cancer cells, cancer stem cells, are more malignant than others. And with that, I would like to thank the patients who donated their tissues for our research, our collaborators, Min City Clinical Oncologic Dispensary, Belarus State Medical University, and International Sakharov Environmental Institute of Belarus State University. Of course, our funders, Belarusian Republican Foundation for Fundamental Research. Of course, I would like to thank Professor Skvartsova and the organizing committee for inviting me to speak at this important conference. And I would like to thank all the audience for their attention. And I would be happy to answer your questions. So many thanks. Bye.
Thank you very much, Marina, for this very interesting presentation. There are quite a few questions in the chat, so maybe I will read them for you. First is from Tihomir Todorov. Do you distinguish between the different isoforms of the CD44 in your staining, as it is shown that only some of its isoforms are associated with the cancer stem cells? And another question is that CD133 is shown to be also expressed on normal prostate stem cells. In this matter, how are you sure that CD44 and CD133 positive cells that you isolate are actually cancer stem cells?
Okay, dear Tihomir, many thanks for your question. So regarding your first question, no, due to, well, technical and financial issues, so in our project we were aiming to stain only standard isoforms of CD44 molecule, and by now we are staining only standard variant of CD44. But yes, you're absolutely right that some isoforms are more associated with CSCs, especially CD44V6, and we are thinking of applying for another grant maybe to do more research on this isoform. And regarding your second question, yes, the phenotype of normal prostate stem cells and prostate CSCs is mostly the same, CD44 positive, CD133 positive. Then we think that those cells are cancer stem cells because we are analyzing tissues. We don't isolate those cells on flow cytometry or any other method. We're using host slides. We do not even use tissue microarrays to address possible tumor heterogeneity on one slide. So we are using slides, and we just see that those cells are located in cancerous areas. So I'm a pathologist. I just can distinguish cancer from normal cells. So that is how I distinguish they are cancer stem cells.
Okay, thank you very much. And there is also one question from Ira. Did you observe any plasticity in CD44 isoforms? Are they stable in prostate cancer cells or could demonstrate different expression?
Many thanks, Professor Skvartsova, for your question. Again, in our research, we didn't address this question, but from literature, it is known that some isoforms may be re-expressed or down-regulated during tumor progression. And as CD44 is regulated by various oncogenic pathways, it has also been shown that some other forms can be upregulated during oncogenic signaling. So CD44 isoform profile, let's say so, may change during cancer cell progression, and different expression, yes, I think it can be demonstrated.
Thank you very much.